Thanks, Alon. Have you ever wondered how new chemical compounds are discovered? Maybe you picture a scientist, like this, doing lots of complicated reactions. Probably some new compounds are discovered this way, but let me introduce you to one of the chemists that I work with the most. Meet Pseudomonas protogens. It's a bacterium. Bacteria are actually amazing chemists. They can produce all kinds of chemical compounds in response to the environment and to help them survive. Many of these compounds have bioactivity, some sort of interaction with living cells, whether beneficial or detrimental. This makes them a really good source of potential new pharmaceuticals. In fact, 2/3 of all new drugs approved by the FDA in recent years have been based on natural products, compounds made by bacteria and fungi. So what we want to do is identify new compounds from bacteria and test them for bioactivity to see whether they improve or inhibit growth of bacterial cells or fungal cells or maybe even human cells. So how do we do that? Thanks to recent improvements in DNA sequencing technology, we now have access to databases filled with genetic information on thousands or even millions of bacterial strains. Within these databases, we can find biosynthetic gene clusters, effectively blueprints encoding the machinery to make these interesting and potentially useful compounds. In many bacterial strains, we can find way more of these clusters than we can find products. Just because a bacterium can do something doesn't mean it always will. Relatable, right? However, this makes it very difficult to figure out what these compounds are and what they do, which is a problem for those of us who work in natural products drug discovery labs. The way that I solve this problem is with strain engineering, which is genetic manipulation to develop a strain of bacteria for a specific purpose. In my case, my purpose is to produce enough of a target compound for structural identification and bioactivity testing. So for example, you might remember my chemist friend from lab, Pseudomonas protogens. It has the same gene cluster as another bacterium. The genes are just in a different order. In the other bacterium, we know that this gene cluster is for a compound called toxoflavin. So you would think that Pseudomonas also produces toxoflavin. But it doesn't, or at least not enough for us to find it in lab. Toxoflavin is an interesting compound for a number of reasons. It is a natural product of many different bacteria. It is also bioactive, and it has different bioactivities in different biological contexts. So for these reasons, we want to discover more compounds that are like toxoflavin. We can look through the databases of bacterial genomes and search for similar gene clusters, blueprints that call for using some of that same machinery that's used to make toxoflavin. However, even though we want to know what these new gene clusters make, we don't just want to grow up the bacteria that we find the gene clusters in because we know that bacteria don't always produce things, even if they have the gene clusters for them. So instead, what I want to do is use Pseudomonas as a little tiny factory to make these new compounds for me. So the top cluster, pictured here, is for toxoflavin. That's from Pseudomonas. The other gene clusters shown here share the same core genes, which are color-coded. They also have some genes which are different in gray, and included are for what we call decorating enzymes. So we hypothesize that all of the compounds made by all of the gene clusters here share the same core chemical structure made by those core genes. They just have different decorations off of that shared intermediate. So for toxoflavin, our decorating enzyme is called TOXA, and we predict it's responsible for these two final methylations. My professor and our collaborators engineered a strain of Pseudomonas that can overproduce toxoflavin, which I used as my basis. Then I knocked out TOXA, so instead of making toxoflavin, my new strain of Pseudomonas produces this intermediate, which is that shared core chemical structure. And so from there, I just needed a way to put the new gene clusters in. Essentially, I added 2 landing pads into the genome, which allow for the integration of new genes in between them, effectively letting me plug and play with the other gene clusters. From there, it's up to the bacterium to synthesize the compounds. I know I make it sound like they do most of the work, but trust me, I have to do work too. Last week, I pulled probably 60 hours or something in lab, and I did it just for you, audience. So I could get this cool data to show you. Thank you. You're so sweet. I know that this is a mess. Don't worry. This is where my work comes in, right? So instead, we're going to ignore all of this extraneous data, and we're just going to focus on these 2 peaks, OK? So we can see that the Pseudomonas strains containing these 4 other gene clusters produced something. That's what these peaks are. And we know that it's something different from toxoflavin, because the Pseudomonas strain containing the toxoflavin gene cluster is that flat line across the bottom in both of those graphs. So it worked. We did it! I don't know what it is yet. I've got as much idea as you do. So now it's time to do the structural identification and the bioactivity testing. Truthfully, I'm not going to be the one who does the structural ID. That's going to be some other grad student in lab, because that's not really my specialty. I'm a bioengineer, not a chemist. I prefer to leave the complex chemistry, whether it's structural ID or compound synthesis, to the experts. Thank you for listening.